Welcome to AP Biology. This video is on meiosis and sexual life cycles. The human life cycle is the alternation of having and doubling of chromosome number that takes place in each generation. Human beings are multicellular diploid organisms. To be diploid means that all your chromosomes exist in pairs. Human beings have 23 pairs of chromosomes, or 46 chromosomes total. Multicellular diploid organisms are going to go through the process of meiosis. Meiosis takes place in the gonads, the male testes or the female ovaries. Meiosis is going to cut the chromosome number in half and is going to create haploid gametes. Haploid gametes have half the number of chromosomes. The male testes produce sperm, the female ovaries produce ova or eggs. The sperm is going to fertilize the egg, which is going to double the chromosome number, creating a diploid zygote. That diploid zygote is then going to grow through mitosis into another multicellular diploid adult. Meiosis cuts the chromosome number in half. The process of mitosis is the process by which organisms grow, and the chromosome number is going to remain the same. Meiosis has a second goal in addition to cutting the chromosome number in half. The second thing we're going to see take place during meiosis is that genetic variation is going to be created. Genetic variation is crucial for the process of evolution. Two things that are universal to all sexual life cycles are meiosis and fertilization. Meiosis cutting the chromosome number in half, fertilization restoring the chromosome number. Some organisms don't reproduce sexually. In sexual reproduction, the male sperm is going to fertilize the female egg. Some organisms reproduce asexually through the process of mitosis. This is going to produce identical clones. Here you see a hydra forming a bud through the process of mitosis, and that bud is going to grow into a new hydra that's identical to the parent. Redwood trees also have the ability to reproduce asexually. Here you see a fallen redwood, and all these trees around it actually form from buds off of the original redwood tree. Each of these trees would be a clone of the original. However, the important thing is that with asexual reproduction, no genetic variation is created. Somatic cells are body cells, and all human somatic cells have 46 chromosomes. They are all diploid. They have 23 pairs of chromosomes. To be diploid, we say that you are 2n, where n is the number of chromosomes. 2 because you have two sets of those. So in human beings, n equals 23, or we have 46 total chromosomes in a human somatic cell. These 23 pairs are called homologous pairs. Homologous means that they contain the same genes in the same locations. You inherit one set from your mother, you inherit the other set from your father. A karyotype is a display of all 46 chromosomes. Here we see a picture of a human karyotype, and what you're looking at here is chromosome pair number 1. So this is one chromosome, this is the other chromosome, and you can see that they contain the same genes in the same locations. The location of a gene is called its locus. 22 pairs, you see right here, these are called autosomal chromosomes. And the 23rd pair is called the sex chromosomes because they are going to determine the sex of the individual. XX would be a female, XY would be a male. There is a wide variety of sexual life cycles. The two processes that are universal to each are going to be meiosis and fertilization. The thing that's going to differ between life cycles is the timing. Here you see three different life cycles: the life cycle of an animal, the life cycle of plants and some algae, and the life cycle of fungi. You can see that each life cycle contains meiosis and fertilization. Meiosis cutting the chromosome number in half, fertilization restoring the chromosome number. You can see that each life cycle has a haploid stage where there's one set of chromosomes and a diploid stage that has pairs of chromosomes. What's going to differ between these groups of organisms is what part of the life cycle is dominant, what part of the life cycle is seen more often. In the case of animals, the dominant part of the life cycle is the diploid stage. When we look at animals, they're multicellular diploid organisms. The gametes exist for a very short period of time, so the haploid component of the life cycle is very reduced. If you look at fungi, you see the complete opposite. A fungus is a multicellular or unicellular haploid organism. Fungi are haploid. That's the dominant stage in their life cycle. Fungi do have the ability to fertilize and create a zygote, but that zygote immediately goes through meiosis, returning it to the haploid state. Fungi do this for one reason. Meiosis creates genetic variation, and genetic variation is crucial for the process of evolution. Plants are kind of unique because they go through a process called Alternation of Generations. You can see that the life cycle is split right in half, where the haploid part and the diploid part are equal. With plants, the haploid generation is its own individual organism. The diploid stage is its own individual organism. So if you look at plants, each generation they alternate. The one generation has a haploid organism, the next generation has a diploid organism. The next generation has a haploid organism, the next generation has a diploid organism. The diploid organism, which is a multicellular diploid adult, is referred to as the sporophyte. The sporophyte, since it's diploid, goes through the process of meiosis and creates haploid cells. These haploid cells are called spores. That's why we call it the sporophyte. Spores don't fertilize each other, which is why they're not gametes. Instead, the haploid spores grow into, through mitosis, a multicellular haploid organism. We call this organism the gametophyte. The gametophyte, since it's haploid, can create gametes through the process of mitosis. That's why we call it the gametophyte. Male gametophytes are going to produce sperm. Female gametophytes are going to produce egg. Sperm's going to fertilize the egg and create a diploid zygote. That begins the next generation, the diploid generation. So plants are unique in that the life cycle is completely split in half. And each generation, each side, the haploid side and the diploid side, has its own individual organism. We call this alternation of generations. The process of meiosis has two goals. One, to cut the chromosome number in half to produce haploid gametes, and two, to produce genetic variation. We're going to see how both of these are accomplished. Interphase is any time when a cell is not dividing, not going through the process of mitosis or not going through the process of meiosis. During interphase, cells are diploid. Meiosis is broken down into two large stages, one called Meiosis 1, one called Meiosis 2. The major event that's going to happen during meiosis 1 is that the homologous pairs are going to separate. So during meiosis 1, the cell goes from being diploid, having pairs, to haploid, now having individual chromosomes. During meiosis 2, the sister chromatids are going to separate. A sister chromatid is each side of this homologous chromosome. So this would be one sister chromatid, this would be the other sister chromatids. Sister chromatids are exact copies of each other that were created during the process of DNA replication. Here we look at the events taking place during meiosis. We begin with meiosis 1. Here we see the cell during interphase. Notice that the chromosomes are not visible. The DNA is found within loose open chromatin. During prophase 1, a number of events are going to take place. 1, the nucleus disappears. 2, the chromatin condenses to form chromosomes. 3, the spindle fibers form. And 4, and probably most important, is that the homologous pairs of chromosomes come together and overlap. In this diagram here, we see two homologous pairs that are coming together and overlapping. During prophase 1, a process is going to take place called crossing over. During crossing over, these homologous chromosomes are going to exchange genetic information. If you look, they are actually flip-flopping their pieces right here. This is going to mix up all the genes. It's going to increase genetic variation. During metaphase 1 of meiosis 1, the spindle fibers attach to the kinetochores of the chromosomes. The homologous pairs line up at the metaphase plate, and that's the key thing to understand there. The pairs line up at the metaphase plate. During anaphase 1, the homologous pairs migrate to either side of the cell. The homologous pairs separate. So you can see that during anaphase 1, that chromosome number is beginning to be cut in half. This cell here begins with 4 chromosomes. At this point, you see 2 chromosomes are going to each side. During telophase 1, the nucleus reforms. During cytokinesis, the cell is going to split in half. We now have 2 cells that each have 2 chromosomes. So during meiosis 1, we went from being diploid to haploid. 4 chromosomes or 2 pairs to now having 2 individual chromosomes. Meiosis 2 is identical to mitosis, except that you're working with half the number of chromosomes. The key event that's going to take place is that during metaphase 2, which is what you see right here, during metaphase 2, the individual chromosomes line up at the metaphase plate. During anaphase 2, the sister chromatids are pulled apart. Sister chromatids get pulled apart. You can see that each chromatid looks different because of that crossing over that took place. During telophase 2, nucleus reforms. Cytokinesis, the cells split in half. You began with 1 diploid cell, and we're ending with 4 haploid daughter cells that are each unique. They all have different chromosomes because of that crossing over that took place. This is going to increase the genetic variation, and these 4 cells are going to become gametes, either sperm or egg. Here we see a comparison between mitosis and meiosis. The major thing you need to understand is that in mitosis, identical copies are made. Mitosis is used for an organism to grow or to repair. Meiosis has a different goal. Its goal is to cut the chromosome number in half and create genetic variation. It's only going to occur in one part of the body, either the male testes or the female ovaries, and lead to the production of gametes. So how is this genetic variation created? The original source of genetic variation are mutations. Mutations are random changes that take place in the genetic code due to mistakes during DNA replication. Mutations are important sources of variation because mutations lead to the creation of new alleles, different versions of genes. However, most genetic variation that takes place is not due to mutations, but is due to the shuffling of genes and the shuffling of alleles that takes place during meiosis. We call this shuffling of alleles genetic recombination. And genetic recombination is the source of most variation that takes place during meiosis. There are three forms of genetic recombination. Number one is called independent assortment of chromosomes. When the homologous pairs line up at the metaphase plate during metaphase 1, nothing dictates which chromosome lines up on which side. Here we look at an example with two pairs. You can see that in this example here, the blues line up on the left, the reds line up on the right. It can just as easily be flip-flopped. In this one here, you have the blue on the top left and the bottom right, the red on the top right and bottom left. It could just as easily be flip-flopped as well. So with two pairs, there is 4 different combinations, right, or 2 to the second power. For 3 pairs, you have 2 to the third power, which would be 2 times 2 times 2, which would be 2 times 2 is 4, 4 times 2 is 8 different pairs, 8 different combinations. 4 pairs is going to have 2 to the 4th power. And you can see that with 23 pairs, there is a ridiculous number of ways that the chromosomes can line up during metaphase. Each unique combination of chromosomes is going to lead to a unique combination of chromosomes and therefore a unique combination of alleles inside the daughter cells. This creates tons of genetic variation. Each gamete, the sperm or the egg, can have 8 million different combinations of alleles, or 8 million different combinations of chromosomes. That would be 2 to the 23rd power since there's 23 pairs. Nothing dictates what sperm fertilizes what egg. We call this random fertilization. So each zygote that results from the fertilization of sperm and egg would be the combination of 1 in 8 million chances times 1 in 8 million chances. So each individual is the result of a 1 in 64 trillion different chances. There are 64 trillion different ways that your mother's chromosomes could have matched up with your father's chromosomes. You can see here why they truly call it the genetic lottery. No two individuals on this planet can ever be genetically identical. The third and final way that genetic variation is created is through crossing over, which we talked about. Crossing over is going to occur during prophase 1. Some books might say that it occurs during metaphase 1. It doesn't really matter when it happens. Here you see a picture of crossing over taking place. When the two homologous pairs are overlapping, we call this entire structure a tetrad. The exact region where the crossing over is taking place, we call this the chiasma or the chiasmata. That's the region where the exchange is actually taking place. You can see here that when they exchange pieces of the chromosomes, when the homologous pairs pull apart, they will have exchanged pieces of their chromosomes. During Anaphase 2, we see the sister chromatids get pulled apart, and if you look at the chromosomes that are inside each gamete, each chromosome is going to be unique. So we have three forms of genetic recombination. One, the independent assortment of chromosomes that takes place during metaphase 1. 2, we have crossing over that takes place during prophase 1. And 3, we have random fertilization. These are the three components of genetic recombination, and genetic recombination is how most genetic variation is created. Variation is at the heart of Darwin's theory of natural selection. Without variation, evolution cannot take place. This concludes our video on meiosis and the sexual life cycles.